Hello, everyone, and welcome to today's session of Global Immunotalks. Now, today we are welcoming Iliyan Iliev. Iliyan did his undergraduate degree in Bulgaria. He then spent some time doing research in Japan before moving to Milan, Italy, where he then did a PhD with Maria Bressigno. After that, he moved to California and was a postdoctoral fellow for some time at Cedars-Sinai Medical Center in working with David Underhill before eventually moving to Weill Cornell Medical College of Cornell University in New York, where he has been ever since and where he is now an associate professor and is co-director and founder of the Microbiome Core Laboratory. Now, so Ilian's research is focused on the study of the mycobiota in health and disease. So I'm going to repeat that word, mycobiota, not microbiota, although mycobiota is part of the microbiota, as I'm sure Ilian will be telling us in just a minute. It's the study of our fungal commensal organisms. And actually, Ilian's research was the first demonstration of the importance of gut commensal organisms from the fungal kingdoms in driving inflammatory bowel disease. And in fact, he has been systematically dissecting the complex interplay between gut commensal fungi and the host and showing how when the balance is disrupted, it can contribute to a variety of diseases, not just IBD, but as he might also tell us today, a bit of it might also contribute to cancer and possibly even allergy. And I think the current effort in his laboratory is about the unique neuromodulatory and cancer immunity modulating properties of the fungi. So in a minute, I'm going to ask Ilian to deliver his lecture, but as usual in this series, I'm going to start with a personal question before the talk. And so, Ilian, your question is, what inspires you to keep pursuing answers in challenging or unsolved areas of science?
Thank you, Gaetano. First, thank you for having me at Global Immunotalks, and congratulations on running such a impactful and unifying community research series for our community. It's been now, what, like 5 years, and everybody loves this series. So back to your question, I think each unanswered question in biology is part of the puzzle to how we understand, you know, how the body works. So we are witnessing that some discoveries can really change people's lives. And we've been a long time in life sciences, and that's what is now, a dogma. And it's an incredible drive. Of course, it is important to ask the right questions. In my own research, I always opted for areas that are much less known, which required diving into different fields and often changing countries to learn what I thought was important to learn. That's a fulfilling but humbling experience. Many times you just feel you go back to first grade. But that's the way how it works. That's how it worked for me. It's the way to push the boundaries, both personally and in our research. We also do translational research, so with the hope we can change some patient lives, which is another drive. And of course, huge inspiration is my team. It's so fulfilling to just go to work and discuss common topics of common interest with other people that are in your team and with inspiring colleagues and collaborators. So that's extremely fulfilling, since we moved to New York, and it's exactly now 10 years. It's been full-time work on the microbiome, so I don't have defect in my speech. It's the microbiome, as you said. So on microbiome and immunity, and it's been extremely fulfilling. I learned a lot from fungi. So I hope I can transmit my excitement today to the audience with my talk. So with that, I will share my screen.
So Thank you, Iliyan. And we look forward to your talk, which is entitled An Immunological Perspective on Gut Fungal Symbiosis from Tissue Homeostasis to Pathology.
Yes, so does everybody see my screen now?
Yes.
Yeah. Okay. Uh, so in our lab, let me just move this slide. So in our lab, we are really interested in how mucosal immunity works. Some of the major questions are how basically the mucosal immune system, which is there to basically sense and interact with the outside environment, and how it will basically respond to harmful organisms and how it will tolerate and choose what to tolerate, which is not harmful. And then once it induces a response, how it will basically suppress that response when it's needed. So it's a lot of decisions that have to be made in a milliseconds. So we know the parts of that the immune system has to interact. There is a fungal component, so fungi are part of the air we breathe and all our environment. They've been part of our diets for millennia. People have been fermenting and cultivating fungi, so our gastrointestinal tract has met fungi on a daily basis for millennia. They're part of our microbiota, and they can also lead to devastating fungal infections in people with suppressed immune system. So a fungal microbiota, or what we call the mycobiota, covers all the body surfaces, and those communities are extremely responsive to pathophysiological conditions. Uh, Fungi have co-evolved with animals since diverging from plants, so that's estimated to be 1 billion years ago, which is plenty of opportunities to co-evolve with the animal immune systems. Uh, if we think of you know, how immunity handles fungi, 97% or even up to that of species on Earth handle fungi exclusively by their innate immune system. And very small percentage, humans included, have also adaptive immune system. So fungal infections have been around a long time, and fungal immunology in its more rudimentary form have been around for a long time, but it's actually very recent. It's about 30 years ago that the tool-like receptors were discovered, and this is one of the landmark studies by Bruno Lemaitre and Jules Hoffman, showing that knocking out tool-like receptor in a fly leads to deadly fungal infection by Aspergillus fumigatus. Another landmark study by Ruslan Metskitov and Charles Genway was the cloning and the discovery of the human TLR. And Ruslan demonstrated that this leads to production of inflammatory cytokine, but importantly, it also drives an adaptive immune response. And my mentor, David Underhill, when he was working with Adam Adhirem, also showed that in the phagosome, when fungal zymosan goes there, and is recognized by tool-like receptor 2. Tool-like receptor 2 can actually distinguish between initiate inflammatory response against the zymosan, so basically distinguish between the fungal and the inert signal. So that's very important. But the field of fungal immunology really exploded with discovery that Gordon Brown and Simon Gordon made, that the C-type lectin receptor lectin-1 is a receptor that senses fungal cell wall beta-glucan. And that opened another big area here, and many labs contributed, and downstream of that signaling, the discovery of card9 in the induction, and how card9 induces basically the adaptive immune response, first by Jürgen Ruland, and also by Caetano's lab, and later also shown that the suppression that Th17 cells are very important for control of fungal infections in the oral mucosa by Sarah Gaffen. And at the same time, many groups were working on other C-type lectin receptors that also go through this signaling, but they use the common gamma chain receptor to phosphorylate Sig, and also the inflammasome was also discovered to be involved in this process. So it has been very rich field since then. And the group of Mihai Netea now little bit more than 10 years ago showed that there is also such a thing as a trained immunity that independently an adaptive immunity can basically preserve some memory. So there actually fungi, Candida albicans was used as, and glucan particles were used as tool to discover that. So, the fungal field is very exciting. But when we think of antifungal immunity, there are unique challenges that it has to basically face compared to antibacterial immunity. First of all, fungi are eukaryotes, so it's very hard to actually target them because we are also eukaryotes, and new antifungal drugs shouldn't be toxic to us. So that's why there are very few antifungal drugs. The fungal cell wall is sugar rich, and on top of it, some fungi such as Cryptococcus, for example, can make a capsule. Many fungi have melanin, so all that shields fungi from the immune system, which is challenging for the immune system to actually recognize them on time and respond. And then the fungal cell size, fungi are much bigger than bacteria, and they can switch between yeast and hypho forms. So it basically is a big challenge for the immune system to really face such a giant cells. So that's why fungal infections and now with immunosuppression become more and more common, unfortunately. And recent estimates show that 2.5 to 3 million fungal infections lead to mortalities per year, which is a huge number. And it was just in 2022 that the World Health Organization put basically fungal pathogens in the priority list, which is 3 years ago. So it's very important to think about that. But fungal infections are less common in one part of the body, or at least that's the part where we cannot really look very easily, and that's the gut. And in the gut, fungal commensalism has been described. So now, more than 10 years ago, we described that fungal symbiosis exists there, and we have been working on different methodologies to visualize those organisms, deep sequencing technologies, and computational approaches to really characterize those communities, which brought this idea of the gut microbiome. In the beginning, of course, many labs have contributed to that. It was seen as this mysterious conglomerate. But the microbiome is actually natural part of the microbiome. Fungi, bacteria, and viruses, they are all part of this community. And characterization, of course, of the gut microbiomes is continuing. But what I'm thinking actually to talk a little bit about today is the importance of uncovering guiding principles of how fungi interact with the host in the gastrointestinal tract, which bring some common ideas of how those interactions are happening and hopefully can advance faster efforts in that area. So as anything that you are researching, you need a model system. So our model system is the gastrointestinal tract, where the process of inflammation and tolerance are worked out, how the nature will control them. And of course, during a number of diseases, that balance is disrupted. So it's a very interesting model system. Microbiota, and there is a very big gap of knowledge if you think what cells in the gastrointestinal tract interact with fungi and in the intestines. That was completely unknown some years back. So we decided, okay, first to actually work on that, we have to make some map here and go after specific points in that map. So the map of the galaxy that I used as an analogy in the previous slide. So Irina Leonardi joined the lab, and she started this series of experiments where we will colonize mice with Candida albicans. We'll be looking what type of phagocytes in the gastrointestinal tract are actually responding and sensing fungi, and we will have several readouts: activation of phagocytes, if there is an active phagocytosis, if that response induces Th17, landmark Th17 response, and if there is some consequence to inflammation. So the first finding was that the first cell type, innate immune cell type that interacts with fungi is the CX3CR1 positive mononuclear phagocytes that are lining the intestinal wall. And we basically developed that when the Clarity methodology started, and we could visualize an entire villi here. So you see those cells in green, and Candida albicans here in red that is taken by those cells. Then what we started thinking is how is fractalkine receptor has anything to do with fungal recognition, because it's a chemokine receptor basically. So what Irina found is that the CX3CR1 cells are expressing actually very high levels of a number of C-type lectin receptors, and also TLR2 that I showed in my introduction, and then downstream molecules important for activation of signaling through those pathways, such as SYK and BSL10, MALT1, which are all part of the CAR9 complex, important for induction of NF-kappa beta response and production of inflammatory cytokines to react against the fungal organisms. So Irina did another very interesting experiment. So we expressed of a class 2 in Candida albicans, transferred mice with OT2 cells, very classical model that were CFSC labeled, so we can actually track proliferation. And we targeted basically the CX3CR1 cells with a DTR system. And looked at the proliferation of those adoptively transferred cells and polarization of Th17 cells. And what turned out to be is that targeting the CX3CR1 cells decreases the proliferation of the adoptively transferred T cells and massively decreases basically the polarization of Th17 cells to Candida albicans. And also the same happens if we don't target the entire cells, because there are different things that can happen if you target an entire cell population with diphtheria toxin. But if we basically target SYK, specifically in CX3CR1 cells, the same thing happens. So we concluded that CX3CR1 cells are very important first responders to fungal colonization in the gut. And when we target those cells in urine-colonized mice with Candida and induced colitis using DSS, mice started losing weight and developing inflammation very quickly when they were depleted by those cells. So those cells are protective against inflammation caused by fungi. So in some human GWAS studies in collaboration with Dermot McGovern and Dalen Lee, we also found that loss of function mutation in CX3CR1 in patients with Crohn's disease leads basically to a decrease of antibodies against fungal menon, those so-called ASC antibodies, but would not affect the development of antibodies against bacterial flagellin, which again pointed out to the fungal culprit in this situation in Crohn's disease. So, this and other studies that I didn't present here brought basically the idea that pre-existing disease, if there is basically a fungal presence that can lead to more severe inflammation, and also that was shown in mice that were knocked out for CAR9 by Harry Sokol and Rami Xavier-Slap and many others over the years. So, this idea is now more accepted. So how does the gut fungus really drive this excessive intestinal inflammation? We developed a large collection from fungi in IBD patients. So that was a huge effort by the isolation and patient consent and all that. Marisa Mesquel, Alexa Simon, when they were in the lab, and Xin Li used that collection to basically look how those different strains, and if we only look at Candida albicans, one species here, to simplify, how they phenotypically look. So you can see here that the same species isolated, strains isolated from different patients have completely different phenotype. Some of them will make hyphae, so those are, those invasive structures that fungi use to acquire basically food and to invade. Or some of them will be completely absent or will have very few of those. And, in analogy, it's almost like as different those were phenotypically as the humans they came out from. So this basically led to other assays that Shinley performed in the lab. And one of those assays was looking how basically those phagocytes will interact with fungi if we develop this in vitro system so we can test many strains. And what really appeared here is that all the strains basically diverge in two overarching categories. So one was low damaging, so strains of Candida albicans that fall into this group have a lower ability to induce damage in this in vitro system, and high damaging strains that have higher ability to induce damage in this system. So we went back actually and colonized germ-free mice. So there is no any microbiota, no other fungi, no other bacteria. So we would colonize them with either the low damaging or the high damaging strains. And what turned out is that the high damaging strains had a very high ability to actually induce Th17 response in the gut, but also drive this neutrophilia response in the intestines, which Fiona Powery and others have shown to be actually a culprit in inflammation in her landmark study in Nature Medicine in UC patients. So when we went back and we looked further, it turned out that those low damaging strains, many of them are actually not making much of filaments, much of hyphae, but high damaging strains have that capacity. And then we use CRISPR system to basically target a gene called TFG1 that is important for this switch of yeast to hypho forms. And you see here that when in high damaging strain, EFG1 was targeted, then the high damaging strain didn't actually make hyphae anymore. So we looked actually at the data set that Susan Nobel developed in her lab of Candida albicans that was colonizing the gut, and they use wild type or they use CFG1 knockout. So what appeared here is that in those EFG sufficient strains that are able to make hyphae, there was a factor gene called ESC1 that was highly expressed. So EC1 is a gene encoding for a polypeptide that Julian Naglick and Bernard Hube showed that leads to the processing of this short peptide, which is toxic to cells called candidialysin. So we focus on that and we ask the question, what will happen if we target now not the hypho program, but if we target only EC1 in those high damaging strains? And that's what we did. And what happened was that candidialysin, of course, was not produced, but then those strains where EC1 was targeted, they completely lost their ability to damage epithelial cells or macrophages. And when we colonize such knockouts in germ-free mice, you see the high damaging strain will induce the Th17 response. The strain where we knocked out only EC1 was not able to induce the response with this efficiency. So we concluded that candidialysin is basically an effector molecule produced by Candida albicans in those high damaging strains. And that's one of the culprits that is basically causing to excessive damage in the intestines. So we went back and we looked at the patients in our clinic that we isolated those strains from. And what it turned out, of course, they are in our clinics here in New York. So they're treated. But they are treated with corticosteroids, they are treated with other immunosuppressive drugs. So we factor that in our model. So we will treat mice with DSS so they develop this severe colitis, but we will treat them with corticosteroid, prednisolone or not, and we'll colonize them with fungi or not. And what turned out is prednisolone, of course, will have some effect. So I'm actually I should have put here the control, but this is the prednisolone treated group, so there is actually less inflammation in that situation. But if Candida was colonizing the mice, irrespective of the treatment with prednisolone, the length of the colon was shortened, and those mice experienced more severe inflammation. So Candida basically in that context was a factor that was inducing more severity and non-response to this medication. So we performed other assays where we were looking what actually is produced when Candida is causing damage of macrophages and IL-1 beta shined. So here each dot is basically a strain, and here it's one assay. So there is a very big correlation, but not with TNF alpha in that situation. So what we did was to basically treat those mice with anti-IL-1 receptor or isotype control. And this treatment basically led to a decrease of the inflammation, telling us that IL-1 receptor, IL-1 pathway is important in this context. And the same happens if you measure the Th17 cells. And in another study that Sarah Gaffin's lab did in the oral mucosa, IL-1 was also very important for the induction of the Th17 cells. So it's kind of probably a conserved phenomenon how this cytokine is important in different contexts, but here in the context of inflammation, that's actually very deleterious. So in conclusion of those findings here, we concluded that some patients carry those fungal strains that produce this toxin called Iliyan, and this toxin is basically targeting the epithelial cells, the underlying macrophages, and that leads to more severe damage in IL-1 dependent mechanism. So, the other very important for us conclusion is working on the microbiota is that the function, but not the composition of the microbiota determines its effect on mucosal immunity and intestinal disease. And we did some correlation studies as well, and actually, these functional studies were telling us where we have actually a phenotype. So I think this is a very important takeout message of this series of studies here. And that broad idea to conclude in this part is that fungi can act in a way as a volume bottom of inflammation in this context. And we saw something similar. Takato Kusakabe did those studies in severe COVID patients that had this very severe inflammation for a long time. The mechanism was a bit different, but fungi in the gut could actually do that. So, fungi in some inflammatory conditions can be this volume bottom to inflammation. And what I'm now excited about, what we are doing in the lab, we are targeting those fungal clones that are expanding in the patients with specific drugs or with microbiota in their metabolites. And we did a lot of preclinical studies, which we completed, and I'll present very soon. But now what I'm excited about is we are actually having ongoing clinical studies at Cornell to really using all those ideas and all those preclinical data and studies that we generated. So this very exciting direction, and in that 10 years anniversary, I'm very happy that the lab was able to go from those basic science questions to really develop those studies and with collaborators to do that. And I will have a lot in my acknowledgement slide you will see later. So in conclusion of this part, we have the microbiome, which is part of the microbiome. And one question that I didn't answer is how the mucosal immune system deals with fungal size, which is not really true, those cells that I have been showing you up to now. So that happened when Itai Duron was in the lab, and he developed this system inspired by studies by Nopalm, where he was looking basically at what are the antibodies binding to fungi, and he developed this system where fungal organisms can be stained with calcafluor white, staining the fungal cell walls, allowing us to distinguish fungi from bacteria, sort them by cells size separation and by antibodies that bind to them. And that was a study that actually brought a lot of very interesting ideas, but I will just go with the main one. In the gut, what we found is that secretory IgAs are actually binding to Candida albicans, but specifically to its hypho form. And here the hypho form is in red. This is with human material here. And the same in mice, where we developed this strain where hyphae, when there is a switch from mist to hyphae, there is a report for RFP. And what is the meaning of that? So we colonize mice, wild-type mice with this reporter so they can make antibodies, and we use mice that cannot make antibodies. And what Itai found was that in presence of antibodies, of course, candida will switch from yeast to hyphoforms in the gut, but that will be dramatically increased in absence of those secretory antibodies, which led us to the conclusion that maybe those antibodies are the one that control the switch to some extent and deal with those huge hyphoforms. And this is, the conclusion of that part. And I want to give a shout out to another study by Kyle Laos and Joe Round, which found the same phenomenon. All those were published in a few years back. So that's very exciting. So I told you about our lessons from disease, but of course we want to learn from health because that's where actually everything important is happening. The disease is the dysregulation. So what Itai also found is that intestinal fungi are the one that diversify the systemic antibody pool in humans and mice, and they work in a way as intestinal vaccines, if you can call it like that, natural vaccine. And he found that this is true CAR9 expressed in phagocytes in the germinal centers. So I will not go through that study, but this is one of those beneficial effects that we think fungi actually, intestinal fungi contribute to humans and mice. And in experimental studies, he showed that it works as a secondary barrier to this gut-disseminated fungal infection after neutropenia is induced. So neutrophils and macrophages are the first one, but the antibodies can actually rescue as a second guard in that situation. So he uncovered this important mechanism. And something else that I showed you, if we target immune pathways or immunosuppression with prednisolone, all that, in all those situations, fungi actually lead to inflammation. But if we actually colonize mice with Candida albicans, induce DSS colitis, and those mice are immunocompetent, nothing happens to the intestines. The DSS will lead to inflammation, but Candida will not lead to more severe inflammation, which basically brought a few ideas that we wanted to pursue in that situation. So we were like, are some fungal organisms in this context actually beneficial to the host? And here I use this image from Gunnar Hansen's lab to show how basically the healthy intestines look. So there is a production of mucus here, bacteria are in this layer, those are food particles. So you see there this zone here where bacteria do not penetrate. And we found the same is true for fungi. I will show you that data quickly, but that inspired studies where Irina Leonardi actually look what fungi colonize the mucosal layer and what fungi are actually in the lumen. And she found that those are very, very different fungal communities. And if she basically assembles mucosal fungal consortium, she found that it's very immunogenic and immunoprotective. And we use this model that Janelle Iris has been using in her labs. So we would deplete the bacteria, give several doses of DSS, and that becomes very toxic to the intestines, and mice basically start dying in absence of bacterial microbiota. So if we actually colonize the mice with the mucosal community, we see a protective effect. If we colonize them with the luminal community, which is probably more of passenger fungi, there was no effect. And she showed that this is true at IL-17 and IL-22 that are protecting the barrier, and they are induced by this mucosal community. And that can actually influence the permeability in the intestines. So she also found, and that's a bonus effect, I think of it as such because we are not a neuro lab, but what she found is that IL-17 produced by those cells can actually act also on neurons through IL-17 receptor and affect social behavior. So that's another third positive effect that we found here. So together, that kind of brought us to the idea and the question, what are the principles that allow those fungal commensals to basically have those positive effects and to persist in the gut, which is a very hostile environment. So, if you go for this question with the systems that we are using now in the lab, so you take basically fungi from the human gut, colonize mice, that's not a system that is adapted because mice don't have actually those fungi. Candidobicas, Candotropicalis, those are not fungal commensals in mice. So we are looking actually at here at this inflammatory response, but we are missing huge here because we are not looking at commensalism, which is a naturally evolved process. So we decided that we need actually evolutionary aligned host where we look at the fungal symbiosis to learn actually how, for the principle of fungal symbiosis and what the host is actually learning from fungi through this evolutionary established relationship. So where can we find a commensal like that? We went into the wild, both in the woods of Maryland, so that was a collaboration with Barbara Reherman, or we went in big cities, and New York City is one of those where we are located. And we could actually collect fecal material from mice in all those locations. And we started thinking, okay, what will be the four principles of commensalism that we have to fulfill to basically say something is a fungal commensal. So in that work, it was more, an overview here and kind of an idea that we developed with wolf fires. So what we thought there is a detection and isolation at scale. Those are principles that have to be fulfilled. There should be proliferation and adaptation to the host. If this is really a commensal, it should withstand competition with the microbiome to live in that niche where it is. And at some point, it should induce probably an immune response. So we collected samples from many different locations on the east, on the west coast, and also in Canada, both from wild mice. And we said, okay, let's go also in different facilities. You never know, although mice in many facilities that we sampled don't have commensal fungi. There is this changing community constantly. But what we found in all those samples was a single organism, single fungus called Kazakhstania tulipazii. And it was shared and it was very similar between the wild mice and some mice that we found in some facilities. So that brought the idea that maybe it has been brought from the wild into some facilities somehow through people walking through, or we don't know how, maybe through food, whatever it is. But it's nevertheless present. So what is Kazakhstania pintulopazia? So it's a part of this Telluris complex that was discovered in 52, and that study described it, and they never went to it. But when the fungus appeared in NIH in 2005, in few mice there was another study that described it. So we thought, okay, that's something very interesting to focus on because it has been seen before. So it's not some, imaginary dream that we found in one study, but what it does. So Iris Gao in the lab, she colonized Jackson mice that do not have it, and they were immediately colonized by this. And when we bred them, they transmitted Kazakhstania in F1. So that stayed for a very long time. Very interestingly, we found that Kazakhstania pintulopazia has this very tight temperature requirement, 30 to 37 degrees, which is very different from Candidobicas, which basically brought us to the idea that probably it evolved very narrowly to actually live in a host because it's very hard to find in nature this type of environment, 30, 37 degrees. And when we actually induce neutropenia in mice where Candida albicans immediately leads to death, because it causes this gut disseminated fungal infection. KP didn't kill its host. And I want to also give a shout out by another study by Stephen Jung's lab that found another species in Kazakhstania, Kazakhstania weizmanni, which is competing with Candida albicans if you co-colonize. So maybe there are a few species of Kazakhstania that are very well adapted to the mouse intestines. So what we described and found is that Kazakhstania is actually living in the gastric mucosa. And you can see here, that's the mucosa here. The mucus is in green, Kazakhstania in red. You can find it in the duodenum, but if you go down in the gastrointestinal tract, in the colon, it's not anymore in the mucus. So it actually created itself a very unique niche. So in that niche, there is no competition with bacteria that you have to withstand in the colon, and it will kind of live there, which is amazing. But what Yun Liao did in the lab, so he used low fiber diet to basically play with the mucus layer, because I showed you that's a very important layer to separate commensals from the mucosal immune system. And what he found in that situation is that with the tining of the mucus, Kazakhstania will induce very strong response, but it was not really the Th17 response, but in the stomach it was, and in the duodenum it was eosinophil type 2 immune response. So that became very interesting, and why it would be happening. What we found is if we use other approaches to really play out with the mucus production, diet, antibiotics, germ-free mice, in all those situations, Kazakhstan will get closer and will induce this eosinophilic type 2 response, which led us to the conclusion that maybe through those fluctuations that are happening normally in life, you always change diets, you always are exposed to things. The mucus is going up and down, right? So, in that situation, fungal commensal can train our immunity, which is also something very interesting. And how it does it, so Yun found that Yun and actually Iris here did this RNA-seq experiment and found that IL-33 is actually produced by the epithelium in that situation. And when Yun targeted IL-33 in intestinal epithelial cells through a genetic approach or through an antibody targeting the receptor of IL-33, ST2, in both situations, he could suppress that using xenophilia, telling us that the fungus is inducing very specific response by the epithelium, and that leads to this underlying type 2 immune response. IL-33, sorry, is a cytokine that has to be activated to actually work. And what Yun found is that Kazakhstania Pentolopeziae can both induce its production in the stomach, but it also can cleave it. And he found that yeast, which, yeast actually don't have digestive system like us inside. So they secrete a lot of proteases, lipases outside to digest material and then absorb the nutrients. So what Yun found is that in this, extract from outside of the fungus secreted fraction, there are a lot of proteases. And when he used different approaches to block those proteases, he found that the serine protease is the one that is actually cleaving the IL-33 in its active form. And now we are investigating that further. So basically, this commensal is using those fluctuation of the mucosal layer to induce an immune response. But would that response matter really to the commensal or to the host? We found that it doesn't matter to the commensal because the commensal colonizes no matter what you do at very high levels. But mice are free-living animals, and they are exposed to parasites, and type 2 immunity evolved to deal with those huge organisms. And what Jung found is, of course, if you colonize mice with Nippel brasiliensis, which is one of those parasites, you get goblet cell hyperplasia, and the parasite, it's a mechanism how the parasite is expelled. But if mice are co-colonized with KP, then there is more of that. And then actually the mice are more protected against the worms. So intended or unintended as a consequence, this can somehow be a protective mechanism to the host. And that's what the fungus is inducing. So in conclusion here, it will be like a couple of big conclusions for everything that I presented. So fungal commensals actively modulate host immunity and shape tissue homeostasis. And what I show today is few examples how we go against towards functional microbiota and towards uncovering mediators and pathways by which fungi actually activate immunity in the host for a good or the worse. And we are under those, following those mechanistic insights. And we are kind of working on the guiding principles of really fungal symbiosis and how that will teach us something about health and disease. There is a disease relevance, as I showed in my initial slides, fungi influence inflammation, and I showed you the data in IBD. So there is this translational developments in the lab that I'm very excited about right now. And what else I'm excited about is what I didn't have time to cover, but those studies hopefully will be coming in the next year or so. Fungi, as I said, they are actually tools to uncover new biology. And we have been finding some very, very interesting uh, phenomenon in the gut-brain axis using basically fungal organisms. We have been working in the area of tumor immunology, which is also very fulfilling, and in the area of cross-kingdom interaction. So fungi and bacteria, they exist in the similar niches. So Will Fires, when he joined the lab many years ago, he started thinking that bacteria should be producing some metabolites that will be basically working against fungi. And indeed, there are such very powerful metabolites that we are working on. And with this, I have to acknowledge many people here, the team, some of the people I told you about during the presenting the slides, but there is a really big team. Some work was not presented here. So I'm really grateful to all the team, to past members that I will also present some of their work. A lot of fulfilling collaborations in Weill Cornell, where we are doing the clinical studies now, and internationally, amazing collaborators from all around the world that we are doing very interesting work together. And of course, that work has been funded by many funders, which we are really grateful for their support. And thank you again for having me here. It has been a great pleasure.
Well, thank you so much, Iliyan, for this amazing talk. Just to remind the audience that Q&A will be in the YouTube channel. You've got a QR code up on your screen right now. And Iliyan will then endeavor to answer your questions in due course. And so it just remains for me to close today's Global Immunotalk. Thank our speaker once again for this
Thank you.
illuminating talk, and remind you all of next week's lecture, which will be by Maria Casanova. And thank you all for continuing to support the Global Immunotalks.